So thank you for coming to my talk. And today I will talk about a work that we did in the last years. The title is Hybridization and Geographic Distribution Shapes the Spatial Genetic Structure of Two Co-occurring Orchid Species. We know that multiple ecological and life history traits shape the fine-scale genetic structure of natural populations. And we can see in this example all this variation may be structured or not, and it depends of the fine-scale conditions of within populations. For example, the occurrence of in core versus peripheral populations can have a huge influence on the patterns of fine-scale structure. And we expect, for example, that in core populations we will have higher outcrossing rates than in peripheral populations, and it depends, for example, can be associated with the abundance of individuals in core versus peripheral populations, the availability of pollinators, and the availability of pollinators may influence the patterns of pollen and seed dispersal in core versus peripheral populations, which will also influence the degree of spatial genetic structure in these populations. And hybridization can also influence the patterns of fine-scale structure. But the influence, curiously, it is hardly being tested in the literature, the influence of hybridization in the patterns of fine-scale structure of populations. So the objective, our main questions and the main objective of this study was to examine the influences of the distribution in peripheral versus core populations and how this differential occurrence influences the patterns of fine-scale structure, the intensity of pollen and seed dispersal in core versus peripheral populations, and the influence of sympatric versus allopatric populations. We know in a previous study that in sympatric populations we have hybridization and introgression. The hybrids are fertile, they can backcross with the parental species, so we recorded interspecific gene exchange between these two species. But we don't know if the hybridization influences the patterns of fine-scale structure within populations. So these are our model species. They belong to the genus Epidendrum. Epidendrum is an orchid genus, the biggest monocot genus with about 1,500 species. And these two species are terrestrial, and they occur in sympatric and allopatric populations along the Brazilian coast. They have very abundant populations, and they occupy this kind of vegetation close to the seashore along the Brazilian coast. It's a tropical environment. Here is a map showing the distribution of the two species, in red Epidendrum punicioluteum and in the other Epidendrum fulgens. Epidendrum fulgens has a bigger distribution, so we tested the influence in core versus peripheral populations using only Epidendrum fulgens because it is distributed, have a bigger distribution. And we used Epidendrum punicioluteum to test the influence of hybridization only in this region where the two species co-occur and hybridize. As I told you, the two species can hybridize and produce a lot of hybrids in the sympatric populations. The hybrids are not as fertile as the parental species, but they can backcross and produce fertile plants. We have gene exchange between these two species. We used 9 nuclear microsatellites and genotyped 581 mapped species. We mapped each individual using a GPS in the populations. We sampled 15 populations, and the extreme populations are 1,100 kilometers apart. And most of the analyses were performed using the software Spaghetti. So one important result that we obtained in this study, using only one species, the species that have the broad distribution, is that the fine-scale genetic structure of the peripheral populations is higher than the ones we found in the core populations. And this agrees with the literature that also expect this pattern, mainly because of biparental inbreeding and lower population densities at peripheral populations. In this kind of situation, we have few individuals in the populations, and most of the